we have had two major developments in the last year or so. One around trying to understand individual driver mutations in cancer and when they occur. So what is a driver mutation? First of all, a driver mutation is a mutation in a gene that alters the biology of the cancer and the way we think about it in a potentially therapeutically tractable way. So where one can use a cancer drug and potentially impact upon the cancer to limit spread or limit its growth for the benefit of the patient. And what we've been looking in human tumours is evidence to see where these particular driver events occur, where and when they occur during the course of human cancer evolution across 11 cancer types. And we published this in Science Translational Medicine earlier on this year. And what we found is that driver mutations aren't always in the trunks of the tumour's evolutionary tree. So you remember from past discussions with you, tumours can be thought of like trees, early events in the trunks, later events in the branches that are present in some cells but not others. And what we're trying to understand is, are there driver events that almost always occur in the trunks, and could we target them more effectively? What we found in a number of cancers is that these driver events aren't always clonal, so they're not always in the trunks of the tumour's evolutionary trees. They're sometimes, or in some cases very often, in the branches. And interestingly, some mutations are more often truncal or clonal than others in particular pathways. So for instance, mutations in the RAS-RAF pathway in human cancer are more commonly clonal, we found, than mutations in the PI3 kinase AKT pathway, which have a tendency to be more frequently branched. And I think that begins to allow us to think about drug development in a new way, that is focusing on tumour events, driver events that are always in the trunk to maximise benefit from targeted therapies. The other thing we found, and the lab is extremely interested in, is what drives branched evolution, because it's this process of diversity in cancers that pharmaceutical companies and clinicians and patients have to battle with, that we don't see in other medical specialties. So we were just discussing briefly the cost of cancer care, but the question is, why is drug development so much more expensive in cancer than it is in other fields of medicine? And one of the reasons for this is this issue of genome instability that we briefly discussed before, this ever-evolving, constantly changing, constantly adapting cancer genome that you don't see in other medical specialties. So we're dealing with a constantly moving target, and what the lab is interested in is identifying how that target changes, how the cancer cells adapt, how diversity ensues in a human tumour. And what we found in a couple of papers are two new routes through which that can happen. The first is through an enzyme that the tumour induces in its genome called APOBEC, which is a cytosine deaminase. What is a cytosine deaminase? Well, it's an enzyme that normal human cells use to fight viral infection by mutating viral DNA and rendering the virus not fit for purpose. Unfortunately, cancers appear to switch on that enzyme in a way in which that enzyme can then mutate the tumour DNA and allow the tumour DNA to mutate and create diversity. And we see this enzyme being induced in the branches of many human cancers, including lung cancer, some breast cancers, bladder cancer, and head and neck squamous carcinomas, as a route to generating diversification. Now, of course, that opens up some very intriguing therapeutic possibilities. If one could inhibit the APOBEC enzyme, could one then begin to limit tumour evolution, limit tumour diversification? And Paul Workman at ICR and I are working on ways in which we can potentially inhibit that enzyme one day for therapeutic benefit. And then the other area that we've been looking at is in the context of evolution within esophageal carcinoma during chemotherapy, to examine what the impact of chemotherapy is upon the cancer genome and how the cancer genome evolves in a very short space of time, in 3 to 4 cycles of platinum-based chemotherapy in esophageal carcinoma. And what we found is that in residual disease, in patients who've been exposed to even just a very short 9 to 12-week block of platinum-based chemotherapy, there are what we call subclonal or branch mutations that we think are new, that have been induced by the chemotherapy. So the chemotherapy is mutagenizing DNA, and leaving a relic, if you like, of tumour cells that have new mutations that potentially could be the substrate for further adaptation and selection during the disease course. So the very drugs that we're using in the clinical setting are inducing mutations that may be the substrate for further selection and adaptation as the tumour grows.